咁但係你話佢哋係執緊垃圾咧，我都覺得係冇乜違和感嘅。今次係考畫圖，下次可以點樣做呢？下次可能已經係俾晒答案你。
Yeah, boy.
咁多位大家好，我係梁 Sir。今日咧，我哋又嚟做一條有關於生態研習嘅題目啦。咁考評局咧，我都幾跟上潮流喎，用 AI 嚟出題目。咁啊，用 AI 咧去生成一個圖像啊。咁佢就落咗一個提示啦，就講啦，一張咧拍攝咗香港學生去岩岸進行實地考察嘅照片，去研究下沿岸生物嘅分佈同埋數量。咁啊得出咗以下呢幅圖畫啦，咁啊幾好啊，山明水秀係咪？咁啊睇得出啦，係一個岩岸嘅位置啦。咁啊當然有個箱啊，綠色呢啲唔知咩嚟啦，綠色石頭啦嚇，咁啊可能係啲膠樽啦係咪？咁佢哋咧就喺個岩岸邊咧就有探究咯。咁其實有關於岩岸嘅生態研習題目咧都唔係第一次出㗎啦。咁啊快手睇下片溫下書咯。Part A 佢就講啦，其實呢張相咧係唔能夠完全反映到呢一句句子嘅要求嘅。呢句句子有咩要求呢？就話呢班學生去岩岸進行實地考察啊嘛。你話喂，咪岩岸咯，咪實地考察咯。咁但係你話佢哋係執緊垃圾呢，我都覺得係冇乜違和感嘅。咁點解呢幅圖啦就好似跟唔到呢個指示去生成呢張圖片呢？咁啊因為啦有兩個好必要嘅工具係冇咗喎。咁所以啦，我哋望翻啦，嗱喺岩岸啦嚇，啲手套啊，有個箱咧去擺啲樣本啊，都可以接受嘅。咁你係咪真係要收集樣本先？定還是個實驗探究係研究緊另一樣嘢呢？原來就係研究一下沿岸嘅生物嘅分佈同埋數量啦。咁呢個題目啦，梗係考翻我哋有關於生態研習嘅基本設備，究竟佢哋帶漏咗啲乜嘢呢？咁自不然就係樣條同埋樣方啦。咁喺過往岩岸嘅生態研習嘅題目咧，我都同大家溫過一次書噶啦。咁我哋咧一般啦，就將個樣條咯同埋樣方啦，就夾埋一齊嚟用啊，就係個樣帶嘅方法啦。我哋就係將呢條樣線啦，由岸嘅一邊拉到去岸嘅近水嘅位置啦，然後啦就將個樣方咧就係連續地或者按住一個特定嘅間距底下咧，就擺喺個樣條嘅一邊或者另一邊，又或者將個樣方嘅中間擺喺個樣條上面，都係可以接受嘅。咁呢個咧就係樣帶嘅方法啦。咁去到第二部分咯，佢就問我哋究竟你係如何點樣去運用翻以上嘅呢個工具樣條咯同埋樣方啦，去收集數據。嗱，呢個咩數據啊？就係去收集翻沿岸生物嘅分佈同埋數量咯。咁留意翻啦，呢個題目咧，其實考生表現又唔係咁好嘅。佢實質要考嘅最關鍵嘅技巧就係系統抽樣法啦。就係幫我哋去研習一下喺一個特定嘅生境入面，究竟啦當中嘅物種佢哋嘅數量啦，同埋分佈嘅。而我哋點樣去運用呢個帶漏咗嘅工具呢？嗱，而家阿 Sir 同你講咗啦，喂，帶漏嘢啊，拎翻嚟俾你啦，你有樣條啦，有樣方啦。第一部分呢唔俾你係冇問題嘅，咪直接喺第二部分講咯。有樣方，有樣條。而家我要去研究沿岸生物嘅分佈同埋數量，你話俾我聽點樣做？其實過往嘅岩岸題目係有問過大家嘅，當年咧就3分，今年就2分。咁我哋睇下個思考應搞咯。今次呢條題目咧，其實就考翻我哋一啲實際嘅操作嘅技巧啦。其實咧亦都等同於我哋嘅 procedure writing，即係話我哋去做 SBA 嘅時候咧，要寫程序步驟噶嘛。第一步做乜，第二步做乜。咁作為一個程序步驟嘅書寫咧，你首先你話俾人哋聽。你用啲咩工具，用咩儀器，用喺啲咩嘅對象身上先？咁呢個係建基於你嘅研究目的。今次嘅研究目的係咩話？就係研究一下沿岸生物嘅分佈同埋數量啦，唔係我吹㗎，題目講㗎咋。第二啦，既然有工具、有儀器、有對象，咁你要有個動詞啦。點樣用嗰樣嘢呢？到第三啦，就係個備註啦。你要去做個步驟，人哋要跟住你呢句去做嘅話啦，擺落去個水浴嗰處幾多度啊？你都要講下啩，係咪？咁所以有關於備註點樣加啲資訊落去，令到你整句步驟係更加完整， 去令到人哋一眼見得到，哦，我知道做乜嘢啦，就唔係話將啲溶液加落個試管，咁加幾多啊？加落邊知道啊？加乜嘢溶液啊？我完全都唔知，我點會跟到你做呢？係咪？咁所以啦，跟翻紅色、藍色、紫色，你就睇一睇咯。第一步，將條羊條放落地下，但係放落地下你就即刻要問啦，備註啦，放喺邊度啊？哦，由個岸嘅尾部去到 waterfront 近水嘅位置，係嘛？然後啦，再將個樣方係嘛放置係嘛？你話其實喂前面唔 lay， 前面都係 place 得唔得啊？可以啊，都係放啫嘛。放置將個樣方咧就放喺個樣條隔離，放喺樣條隔離，可唔可以好似我樓上提大家咁樣款啊？連續地或者一個相對嘅間距上面咧，就 on one side of the transept， 可唔可以？可以。總之你要肯定人哋望到你呢句之後放個樣方點放法，人哋會要知道嘅。跟住啦就數啦，action verb 咯。數咩啊？數下啲物種究竟有幾多款啦，同埋各自嘅數量啦。喂，咁但係咪求其數啊？唔係噶，喺個樣方入面你先係咁樣數噶。所以啦，你就當一個步驟嘅書寫，同過往嘅答案係一模一樣嘅。嗱，今次2024年佢本身嗰個評卷參考咧，咁當然啦，都係嗰句評卷參考只係俾我哋改卷嘅時候作為一個參考，佢唔係絕對嘅答案。所以你話呢個答案，你覺得爭啲乜嘢啊？我就會覺得，喂，不如加埋個喺個特定間距嗰處就放個樣方喺度啦，就唔好淨係話放喺個樣條隔離啦。咁得唔得啊？梗係得啦，令到個答案更加豐富啊嘛，係咪？同埋今次呢個答案咧就係數下啲物種有幾多款，同埋佢哋各自嘅數量啦。但係上次2013年嗰條嘅題目咧就係話啦，我有阿 A 同阿 B 兩款物種嘅啫，即係話如果喺個樣方入面係有物種 C、物種 D 嘅話咧，其實你係可以唔使理會嘅。但係今次唔同喎，係嘛？佢話喺個岩岸揾啲生物啫嘛，佢幾多款生物你又唔知，係嘛？啲生物嘅數量你又唔知，咁你咪話俾人哋聽咯。物種種類嘅數量同埋各自物種所有嘅數量咪已經 OK 咯。咁亦都借呢個機會講下一啲常犯嘅錯誤啦。第一類型嘅常犯錯誤咧，就係用英文答題目嘅同學咧，就將呢個 line transept 樣線法同埋 transept line 樣條或者樣線咧，就係撈亂咗。而家你問緊嘅係嗰個工具，個工具咧係樣線，啱唔啱？但係樣線法 line transept 咧就係嗰個方法嚟嘅，其實好易記嘅啫。你用英文嘅話，呢個叫 BIO transept 啫嘛，就係樣方加樣條啊嘛。而 line transept 嘅話，咪即係淨係用嗰條樣條去做你嘅探究咯。如果呢個 line 咧，拉條 transept 咪即係手字邊加個立字，拉條繩嗰個意思咯。你可以咁樣去記下嘅。第二類常見嘅錯誤咧，就係啲同學就係答樣帶法啦 ，BIO transept， 因為對於佢哋嘅理解，樣帶咧有樣方同樣條啊嘛，梁 Sir， 咁我咪答俾你咯。但係樣帶法係一個採樣嘅方法，佢唔係工具嚟嘅，嗰個工具應該講翻係樣條同埋樣方先真嘅。咁同埋啦，有好多同學咧就講咗啲無關痛癢嘅嘢啦，用溫度計啊、間尺啊、風測儀啊、量度下嗰個水嘅 pH 啊、鹽嘅濃度啊，諸如此類。但係同我哋嘅探究嘅目的咧係冇關係嘅。而家我哋嘅目的係想研究一下沿岸生物嘅分佈同埋數量。咁水溫啊、水入面幾多嘅酸鹼值，其實就唔能夠直接話俾你聽佢哋嘅分佈同埋數量。咁正正就係頭先提大家嘅嘢啦。你要揀啱一個工具，都要建基於你嘅實驗探究目的係咩咯？有關於第二部分咧，亦都有啲同學啦，就淨係講咗數下生物嘅數量，但係題目係話研究生物分佈同數量，其實冇講過淨係研究某款生物。就唔似2013年嗰條，我淨係想研究阿 A 同阿 B， 你咪淨係數阿 A 同阿 B 咯，咁數就可以噶啦。但係啦，而家係講緊呢個岩岸當中有冇其他生物，佢冇透露過俾你聽。所以啦，物種嘅數量啦，同埋每種物種個體嘅數量啦，我哋都要去數清楚嘅。然後啦，去到第二部分啦，咁佢就講咯喎，喺個岩岸嘅呢個探究當中咧，咁有個學生就揾到呢個好薄片嘅啡色嘅一浸嘢喺個岩石上面喎。咁學生就思疑啦，佢會唔會係一啲藻類嚟嘅咧？ 咁佢就拎咗翻去啦，掉咗落去個箱嗰度啦，咁啊拎翻學校咧就做研究下喎。咁題目咧就講啦，用翻咧喺實驗室有嘅一啲儀器啊、溶液啊諸如此類啦，就叫你去設計一個裝置，而呢個裝置係能夠顯示到俾我哋睇，究竟呢一個好薄嘅薄片咧，佢能唔能夠做到呼吸作用咧？然後啦，要你畫翻幅圖喎，好難得咧見到兩條咁相近嘅題目㗎。頭先 A2 咧係要你用文字演繹話俾人哋聽點樣做，然後去到 Part B 咧就要你畫圖去話俾人哋聽點樣做。其實都幾特別地係兩條係極度相似嘅題目嚟嘅。當然啦，個技巧就梗係好唔同啦，係咪？咁當然啦，呢條題目主要就係考下技巧啦。咁但係概念嘅嘢咧都冇得走雞嘅，就係生物嘅7大特性或者7大功能啦。今次就係想考翻呼吸作用啦，係咪？而入翻條題目嘅話，畫個裝置，咁又係考翻我哋 SBA 嘅技巧啦。咁要設計一個實驗，設計一個裝置，又係嗰句啦。你有個儀器，個儀器係同你嘅目的係有關係嘅，就係想睇下究竟呢個薄片係咪真係能夠做到呼吸作用喎？咁其中一個呼吸作用進行到嘅特徵咧，就係製造咗二氧化碳啦。咁喺翻實驗室有冇相應嘅材料、工具、儀器俾你去用咧？就一定有嘅。而當你設計完之後啦，咁你要畫圖喎，係嘛？要有標題啦。今次俾咗你啦，咁下次唔俾噶啦，又睇下幾多同學咧又中招啦。然後畫圖啦，記得畫圖就係畫一個縱切面嘅圖啦，同埋有翻標示啦，究竟你用緊嘅係一個咩嘅工具啦，或者用咩嘅溶液啦。咁今次呢條題目咧，就係相比起2015年嘅呢一條過氧化氫酶咧，就難咗少少嘅，因為佢冇講俾你聽究竟而家你有咩嘅器具喺手，有咩儀器喺手，就純粹就係講實驗室有㗎啦。OK 咁當然啦，你又唔需要用啲好富貴嘅嘢嘅嚇，實驗室有嘅都係以前我哋低年班學嘅啫，係咪？有試管啦，試管加埋個膠室啦，唔同嘅燒杯啊、燒瓶啊諸如此類啦。咁當然啦，呢啲係俾你認下嘅啫，你唔需要用晒㗎嘛。咁亦都借呢個機會咧提下大家啦，畫圖嘅時候咧，用 HB 嘅鉛筆啦，如果要畫直線嘅就用間尺唔好去搽陰影所有嘅工具咧最好就係按比例去畫每一個儀器有翻標示同埋肯定你畫嗰幅圖係夠大先，你唔使畫到蚊枝咁細咁我哋改都改得容易啲係咪？咁對於呢個裝置究竟有啲咩要求咧？第一，佢要係一個閉合嘅系統啦。又或者啦，係一個氣泵嘅系統啦。第二啦，就要阻擋咗啲光嘅。點解會要冇光呢？因為啦，而家我想研究嘅就係呢個薄嘅物質。第一，我思疑佢係藻類，即係話其實佢係有機會接觸到光之後就會做到光合作用。但係我想研究嘅唔係光合作用，係呼吸作用。咁即係話我淨係想呢一片嘅薄片咧進行呼吸作用啫。咁我驚佢做光合作用啊嘛，咁阻擋咗啲光，唔俾曬燈嘅，唔俾棵嘢接收到光嘅，都好正常啦。第三啦，呢個方法要係有效嘅、真確嘅，係話到俾你哋聽個二氧化碳濃度嘅改變啦，又或者啦呢一個物質佢真係製造到二氧化碳嘅。咁睇圖之前咧，就講一講個題型嘅變奏咯。嗱，今次係考畫圖。下次可以點樣做呢？下次可能已經係俾晒答案你，然後就問你啦。喂，喺個實驗之前呢，最緊要就係確保佢係一個閉合嘅系統確保係要冇光確保嗰個裝置係真係能夠顯示得到二氧化碳濃度嘅轉變或者呢個薄嘅物質係製造到二氧化碳。究竟點解要做呢啲步驟呢？又或者啦，好似2017年咁樣，又係俾個裝置你噶啦，就問翻啦，點解當我哋去預備呢個裝置嘅時候，要做以上嘅設定呢？又係嗰啲啦，要係閉合系統遮光同埋點解呢個裝置係能夠顯示得到二氧化碳嘅濃度嘅轉變諸如此類啦。而當年嘅考生表現呢，其實都係非常之差嘅。咁我哋嚟睇下啦，個裝置應該要點樣畫咯？裝置咧有兩大類型，第一類型咧都係利用咗石灰水啦，或者碳酸氫鹽指示劑啦，去顯示翻個二氧化碳濃度嘅變化嘅。咁最典型啦，碳酸氫鹽指示劑正常濃度0.03就係紅色， 高過0.03就係黃色，少過0.03咧就係紫色嚟嘅。咁當中啦，望一望咯喎，首先啦有個塞咁塞住個試管，擺個樣本響樹，咁留意唔好直接浸住喎。有啲同學咧兜嘢滑咗落去浸住咗嘅喎，你浸下浸下會唔會浸死佢㗎？係咪？咁同埋啦，仲有一個錫紙啦，同埋黑紙啦，去做個遮光。跟住啦，又用下唔同嘅裝置咯。咁入氣出氣嘅，咁留意下啦，都要有翻個塞住啦。各自嘅管啦，呢邊插住水嘅就入氣係嘛？鹼石灰係攞嚟吸個二氧化碳嘅。確保進入去呢個樣本嘅空氣咧係冇二氧化碳跟住啦，有石灰水啦，同埋碳酸氫鹽指示劑咧就確保啦入嚟，真係冇二氧化碳嘅喎，可能係紫色啦。跟住過咗個樣本之後出翻嚟，會變咩色咧？會唔會變咗做黃色咧？咁即係話當中係有啲二氧化碳可以產生咗嘅咯噃。咁呢邊就係抽氣啦。同樣地啦，都有翻個黑紙或者錫紙去包住去遮光啦。咁呢個係第一類型嘅裝置。而第二類型嘅裝置咧，就唔係用個碳酸氫鹽指示劑啦，係用個氣壓啦。同樣地呢個大嘅燒瓶啦，咁又係用錫紙黑紙遮住啦，又係將個樣本擺喺度啦。有冇水浸住佢其實冇乜所謂嘅，你咪由佢攤喺度咯。總之就唔好浸住咗呢啲碳酸氫鹽指示劑石灰水就冇問題噶啦。跟住入面咧就照樣擺鹼石灰又係吸咗啲二氧化碳去嘅。呢個樣本吸咗啲氧氣。放咗啲二氧化碳，咁氧氣會少咗，二氧化碳咧就會放咗出嚟，但係又俾個鹼石灰吸咗喎。咁即係話而家呢個燒瓶入面咧，空氣嘅份量係少咗嘅，氣壓應該都係少咗嘅。咁當然氣壓低咗，咁自不然會低過大氣嘅壓力啦。咁所以啦，外面壓力撳落去，咁液面應該係會上升嘅。又講下題型變奏咯喎，今次題目咧就叫你畫圖啦，係咪？下次可以點樣做啊？我俾晒啲裝置你 ，OK？ 我唔理你邊個裝置，1、2、3都好，俾咗你噶啦已經。然後要你指出及解釋我哋嘅可以預期嘅結果。如果呢一個嘅薄片物質佢真係一個藻類嘅話，結果會如何呢？咁最典型啦，阿1同阿2如果佢真係一隻生物啦，佢係真係會進行一個呼吸作用嘅話咧，咁自不然咧個石灰水就會由冇色轉做奶白色啦，又或者個碳酸氫鹽指示劑就會由紅色轉做黃色啦。咁我指出咗啦，我未解釋㗎，你試下解釋啦。跟住啦，第三個裝置咧就係個液面咧係會上升嘅，因為外面啲水撳咗落去啊嘛。咁咪將呢度啲水推咗上去咯。咁但係我又係冇解釋嘅，咁你又可以試喺留言區留低你嘅答案，睇下你 concept 清唔清咯。咁我哋啱啱咧就學咗點樣正確咁畫個裝置啦。咁去到第二個階段就係話，如果我俾個裝置你，要你去評估下佢、評價下佢，或者去改良下佢，即係話你要噏得出佢有啲咩錯咧，係咪？咁我哋又嚟睇下有啲咩好，有啲咩唔好咯。咁我哋先睇呢兩個裝置咯。第一個裝置咧，你見得到啦，同我哋嘅裝置2咧係非常之似嘅。入氣啦、出氣啦、閉合系統啦，加晒木塞啦，係咪？咁呢個 potassium hydroxide 輕氧化鉀咧，其實都係去吸二氧化碳嘅，叫做 lime water 啦、lime water 啦。咁睇下變唔變色啦，係咪？咁呢個啡色嘅物質就擺咗喺呢度啦。咁呢個裝置啦。咁從個裝置嘅基本設計上面咧 OK 嘅。咁衰在佢冇講過係咪成個裝置都擺喺一個黑暗嘅地方去進行，甚至乎外面有冇話擺咗個錫紙或者黑紙咧去遮住光咧，佢又冇講到。咁呢度啦就爭少少啦，所以得2個 tick 嘅啫。跟住去到呢一個咯，望起上嚟都係靚仔仔啦，係咪？用海水浸住都幾體貼啊，係咪？跟住啦，啲二氧化碳出咗去嘅時候啦，又用翻個石灰水去測試下，咁佢就由呢個冇色變做奶白色。咁呢個裝置有冇啲特定嘅缺陷咧？佢又係冇講過，佢要喺黑暗嘅地方進行。咁呢度亦都冇話包住張紙，有一個咩錫紙黑紙咁嘅款，咁所以都係爭少少。咁同埋有啲細位咧，大家都留意下啦。例如啦，呢個出氣位點解會埋口嘅咧？係咪應該開口咧？因為見到啦，啲管開口噶嘛，咁所以你見到就算呢兩個答案，哇，好靚仔都好啦，佢話唔定都有啲錯處，而我哋就要去揾到佢同埋改良啦。咁過往咧都有一條咁嘅題目係問過大家嘅，咁快手又睇下片揾下書咯。仲有冇啊？再睇下啦，例如呢個裝置啦，紅色呢個位打開晒，連個膠塞都冇嘅，係咪？跟住啦， 成個試管咧又冇用嗰啲咩黑紙啊、錫紙包住喎，咁所以啦呢個裝置咧就淨係得個石灰水係有分嘅啫。又或者啦呢個裝置喂好好啊，講到冇光咯喎，係咪？但係啦呢一邊做咩製造氧氣俾氣佢咧？嗱咁你氣嘅流動佢又冇講喎，係咪呢邊入氣呢邊出氣啊？但係呢邊咧又吸咗啲二氧化碳喎，咁一邊製造一邊吸咗佢，咁你用咩辦法去測量到個二氧化碳濃度嘅改變啊？所以你會見到呢個裝置咧根本係冇效嘅，即係啦，好似呢個裝置咁樣啦，嗱好好啊，閉合系統啦，係咪？啊擺晒支管啦，隔離啦就係呢個指示劑啦，還可以接受啊，係咪？咁問題就嚟啦，呢支管又係冇黑紙冇錫紙包住啦，同埋啦另一樣嘅細位咧就係你做咩用啲糖水浸住佢咧？你宜得佢死啊？你唔怕呢個葡萄糖溶液係會抽咗個水藻嘅水分嘅咩？係咪？咁所以呢一 part 咧都係有少少畫蛇添足嘅，所以呢一部分又要小心啲啦。另一個啦，又係望起上嚟，喂好似樣啊！咁首先啦，佢又冇咗個塞啦，所以佢唔係一個閉合嘅系統啦。第二啦，就係個樣本，喂有冇錫紙有冇黑紙包住啊？又冇喎。同埋呢個藻類咧，係咪好似百變怪啊？其實冇乜所謂嘅，你畫圓形啊咩都好，總之你有一個指示咧都 OK 嘅啦。啊，純粹講笑啫，話佢似百變怪。而另外3個樽咧就要留意啦，呢度淨係寫二氧化碳係咩意思啊？吸二氧化碳定係會放啲二氧化碳出嚟？跟住啦就有 hydrogen carbon 嚟去吸二氧化碳，其實就應該唔係啦，應該係講緊 sodium hydroxide 氫氧化鈉或者氫氧化鉀嗱呢類嘅物質先係會吸二氧化碳啦。咁跟住啦，再過到嚟呢邊咧，就係我相信佢想寫石灰水嘅 lime water 啦，我估係。跟住啦，如果呼吸作用發生咗嘅話咧，咁就會變咗做 milky。咁呢個裝置當中其實都符合唔到啲要求嘅，又唔係閉合嘅系統啦，又冇阻擋到啲光線啦。咁同埋最後尾雖然係擺咗個石灰水，但係前面呢啲裝置係不明所以嘅。咁所以啦，大家就要學識去品評一下一個裝置一個設計究竟點樣去改良咯。咁亦都借呢個機會講下啲關鍵嘅技巧啦。例如啦，我哋要將個藻類將呢個樣本咧擺喺黑暗嘅地方咁先。因為啦，如果呢個樣本佢真係個水藻嚟嘅，佢係能夠進行到光合作用嘅。如果有光嘅情況底下，咁就會影響咗佢嗰個正向嘅氣體交換啦。咁所以我淨係想佢做呼吸作用，去淨放出二氧化碳啦，係咪？第二啦，亦都要確保咧，我哋嗰啲接駁管啊係接駁得啱嘅，亦都要用個塞啦，用個 stopper 啦，去固定佢哋嘅位置，去確保啦，整個裝置都係一個閉合嘅系統。如果啲管咧，好似呢個裝置咁樣咧，全部齊口嘅話咧，其實你都唔知究竟係咪要倒落個水度，即係好似呢個咧，其實嗰條管應該插住落個水度嘅。咁你冇嘅話，其實啲空氣流動去咗邊咧，你都不知道。空氣嘅流動會唔會被阻隔咗咧，你都不知道。咁甚至乎，如果佢唔係一個閉合嘅系統嘅話，啲空氣直頭會漏咗出去或者入咗嚟添。咁究竟你係咪測試緊呢一隻疑似藻類嘅樣本，佢係咪真係進行到呼吸作用咧？咁所以過往咧，我哋喺呼吸個課你都有學過嘅。有啲管咧插晒落水嘅，代表住咧啲空氣係進入呢個液體當中。而呢個管係凌空嘅話咧，就係代表啲氣咧係由呢度離開嘅。所以你會發現啦，當初呢個裝置喺我哋呼吸嗰課學過嘅。當我吸氣嘅話咧，係應該由呢一個左邊嘅箭嘴上去嘅，而呼氣係呼落去右手邊呢支嘅試管當中嘅。咁所以你就睇翻嗰條管係咪插落個水度咯。Hello student, I'm Mr. Leung. So this time we are going to do a question about the ecological study. And the HKEA, you can see that they are very trendy. They use the AI as the question. It says that the following photograph was generated by an artificial intelligence program using the following sentence. A photograph capturing a Hong Kong students on a field trip to a rocky shore, studying the distribution and abundance of organisms along the shore.So you can see that in Rocky Shore is a very popular place habitat for us to make the question. So let's take a look at the question. The photograph doesn't truly reflect the requirement in the sentence because 2 pieces of essential equipment are missing. So from the picture, you can see a group of students, the Rocky Shore, and they are really doing something, maybe studying in the Rocky Shore.However, maybe they are just picking up some rubbish from the shore. So, for part A, list the 2 pieces of the essential equipment for the study. So from this question, we need to recognize the missing equipment for ecological studies, and surely they are the transit and the contract. So in the question before, you can watch the video for the revision. So I taught you guys about the belt transit. For the belt transit method, a transit is laid down along the area to be studied, maybe from the back of the shore to the waterfront, and then the contract are placed continuously or at a fixed interval on one side, maybe on this side, maybe on this side, or we place the center of each contract along the transit line. And then for part 2, how could you use the equipment listed in part 1, the transit and the contract, to collect the data needed for the studies? So you can see that somehow if we delete the part A1, I can directly ask you to part A2. How could you use transit and contract to collect the data needed for the studies? And the critical skills is about the basic field trip skills. The systematic sampling method is useful for recording the changes in the abundance and distribution of the species in the habitats, and is what we ask in the past question. Briefly, describe the sampling procedure used to obtain the above data. So how to use the missing equipment? So let's talk about the scaffolding. So for this question, is checking us the practical or the S-based skills. It's about the procedures writing. For the procedures, you need to write down, first step, what do we need to do? Second step, what do we need to do? And in every step, you use a short sentence to present what are you going to do. There are 3 elements you need to put it in. First of all, the apparatus, the tools, the target. They are all related to the purpose. So now the purpose is to study, to investigate the distribution and the abundance of the organism along the shore. Secondly, for the action verb. So you have the apparatus, so you use the action verb, how to use it, or transfer the solution, put the test tube in the water bath, whatever. So the action verb. And for the remarks, it could be about the temperature, duration, location, distance, volume, number of drops, etc. For example, if you say that transfer the solution to the test tube, so I may ask that, what solution? Starch solution. So how much? Or maybe 1 ml starch solution to the test tube. So it can make the procedure much more comprehensive. So let's take a look at the answer. So we laid the transit line from the back shore to the waterfront. So we have the action verb, we have the apparatus, and we have the location. It's much better than you just say that place the transit on the floor. On the floor, which part do you mean? So place the contract next to the transit. So it also tell me the location, on one side. So the contracts are placed on one side of the transit, right? And then for the second step, collect the data. What are the data? Count the number of species and the abundance within the contract. This statement is clearer than I just say that count the number of species and their abundance. Where are the organisms? They are within the contract. So remember that for the procedure writing, apparatus, action verb, and the remarks. So you can see that the answer for the 2013 question. So place the transit line from the back of the shore to the waterfront. Place the contract along the transit line at regular intervals. So you can maybe for this year to transit from back shore to waterfront and place a contract next to the transit at a regular intervals. Much better. And count the number of species A and B in the contract and record the result. But this time, we are not just focusing on species A and B. We are focusing any organism I can find in the rocky shore. Therefore, we need to count the number or species and their abundance. So, for the common mistake, first of all, some candidate taking the English version of the exam paper mix up the line transit with the transit line in their answer. Line transit is the sampling method, and transit line, that's the sampling tools used in the method. It's just like that belt transit is the method, right? line transit is also the method. Some candidates put down BIO transit as their answer without knowing that it was a sampling method involving the use of the two missing equipment, transit line and the contrast. They just mentioned BIO transit only. And many candidates give some general equipment or apparatus such as thermometer or ruler, which are not related to the data collection for the distribution and abundance of the organism along the shore. So read the question carefully. And then for part A2, some candidates simply mention counting the number of the organisms only. They regret that the data collection should involve both the number of species, how many types are there, and then the number of individuals of each species in order to address the study of the distribution and the abundance of the organism referred to in the statement. So one more reminder, read the question carefully. And then for part B, in the field trip to the rocky shore, a student found a thin brown sheet lying on the rock, and the student suspected that it was an algae. So a small pieces of the sample was taken back to the school for further studies. And after going back to school, the student used the apparatus and reagent available in the school laboratory, design a setup which can be used to show whether the sample of this thin sheet can undergo respiration. So surely for this question, it's checking us the skills, the SBA, the practical skills. And meanwhile, it also checks us the concept. It's about the 7 functions of the features of the living things: respiration, reproduction, growth, etc. So now we are checking the respiration. So for the skill checking, it's about SBA skills. It asks us to design the setup and how is the setting. So as what I say before, so as what I mentioned, we need to choose the apparatus wisely based on the purpose. So now the purpose is to show whether the sample of this thin sheet can undergo respiration. And for the respiration, one of the parameters we can use is the production of the carbon dioxide. And for the scientific drawing, we need a title. Luckily, it was given by the question, but maybe next time it's not given. And then let's see how many students forget the title. And then for the drawing, surely we need the vertical section of the apparatus, the test tube, the beaker, the conical vase. So it's all about the skills, about the biological drawing or the scientific drawing. And then for the 2015 question, we also need to design the setup, right? A bit more difficult. It doesn't tell you any hints about the apparatus or the reagent. It just say that using the apparatus and reagents available in the school laboratory. Luckily, we just pick up some ordinary apparatus is okay. Tips for drawing vertical section diagram. First of all, draw with the HB pencil. So draw straight lines with a ruler. Do not shade the diagrams, keep all parts in proportion, label each pieces of the apparatus, and make sure that the diagram is large enough so that each piece of apparatus is clear. So before we talk about the answer, I need to tell you that the basic elements of the setup. First of all, it should be a closed system or an air pump system. And then there should be the blockage of the light. And thirdly, it should include the development method to show the change in the carbon dioxide concentration or the production of the carbon dioxide. So in this question, it asks you to design and draw the setup. So what about possible question variation? It can ask you that the precaution. For example, the setup, the diagram is given. And maybe all these precautions, they are also given, or it's important to block the light. It's important to make the whole system, whole setup as a closed system. The beginning of the experiment, because ... Or all these 3 precautions are given and then ask you that why are above settings necessary when preparing the setup? So you need to be able to explain why I need to do it. And then let's take a look at the diagram. Beware of that. The candidate report the performance of the students are very poor. So there are different types of setup. For the first type, possible setup 1 and 2, this type of setup, we use the lime water or the hydrogen carbon indicator to show the changes in the carbon dioxide concentration or to show that there is production of carbon dioxide. For example, the hydrogen carbon indicator, it shows you that for the 0.03% carbon dioxide, it shows red color, higher than that, yellow color, lower than that, maybe purple color. And for these two setup, it also show you the closed system with the stopper, air pump system, one air in, one air out. And then we also use the aluminum foil or the black paper to block the light. Make sure that the sample suspected it was an alga, so it will not undergo photosynthesis and the sample will only undergo respiration. So it includes all these three elements. And for the second type of the setup, it doesn't talk about the color change of the solution of the reagent. It's talking about the liquid level. So we also use the aluminum foil black paper to block the conical flask, block the light. And then in the conical flask, we use the soda lime for absorption of the carbon dioxide. So in the conical flask, you will find that for the oxygen, the sample will use it, and then it will release carbon dioxide. But the carbon dioxide is absorbed by the soda lime. Therefore, in the air content, the air volume in the flask will be smaller. And then the air pressure in the conical flask will be lower than the atmospheric pressure. So the pressure will press the water will go into the capillary tube, so the liquid level will rise, will go up, right? So this time it asks you to design and draw the setup. So next time it may ask you that all the setups they are given, or maybe setup 1 is given, and then state and explain the expected result of the above setup. If the thin brown sheet is really an alga, you need to talk about that. Or maybe for setup 1 and setup 2, if the thin brown sheet is really an alga, it can really undergo the respiration in the setup during the experiment. So the lime water will turn from colorless to milky white, or the hydrogen carbon indicator will turn from red to yellow. They are the expected results. And for the setup 3, if the thin brown sheet is really an alga, the liquid level will rise. So I state the expected result already. But explanation, I leave it to you. So you can leave your answer in the comment section and let's see your concept is clear or not. So after we talk about the correct answer, so we move on to the next level is that what if the setups they are given, the drawing they are given already in the diagram, and then it asks you to evaluate it, to comment on it. So let's take a look at these two diagrams. So you can see that, for this one, it looks like as the setup 2. So air pump system, air in, air out, and then with the potassium hydroxide, which can absorb the carbon dioxide, lime water to check that the incoming air, there is no carbon dioxide, and after that the brown sheet, and then after that also another lime water. If it really turns from colorless, from milky, so it shows that the change in the carbon dioxide concentration, and it's the air pump system. However, does this diagram score the full mark? No, because it doesn't show you the blockage of the light. There is no aluminum foil or black paper covering the conical flask. There is still room for improvement for this diagram. And for the second setup, you can see that, for the stopper, very good, closed system. And then with the sample in the water, some students, they directly put the sample in the lime water or in the sodium hydrogen carbon indicator. So it's not the valid method. For this diagram, it's not that perfect because the aluminum foil or the black paper are also missing. And then maybe some minor part of this diagram is that why the tube is blocked. The tube should be open, right? So beware of that. So you can see that after reading different types of diagram, we can predict the possible question variation. So you were given the setup already, just like 2023 question. So state any mistake made in the setup and modify the setup. So you can see that. Add the stopper to make a closed system. And for the test tube, surely we need the aluminum foil or the black paper to block the light. So there are two things missing. And then for this part, it really tells you that there should be low light condition. However, for this setup, produce O2, oxygen, absorb CO2, carbon dioxide, and then the alga inside. So for the whole setup, what is the valid method to show the change in the carbon dioxide concentration or the production of the carbon dioxide? It doesn't show it. And then for this setup, you can see the stopper, very good. And then, like before, without the aluminum foil or the black paper to block the light, it's not immersed in the sodium hydrogen carbonate indicator. And one minor thing, why the test tube is broken. It doesn't make sense, right? And you are drawing the diagram. Be careful. And for this setup, one more question is about the glucose solution. Why do you add the glucose solution in the tube? What about use the seawater or water only? Or just put it in the tube? Is it also okay? Because when you put the glucose solution, so maybe the algal cells, the water will be withdrawn by the glucose solution, by osmosis, right? So it will affect your result. And then for this setup, so you can see that no stopper. It is an open system, not closed one. And with the air pump system, so where is the air in? Where is the air out? You did not mention it. And then for the agar, so there is no aluminum foil or black paper to cover it to block the light. It looks like a Pokemon, just kidding. For the reagent, 3 bottle, or for the CO2, what do you mean? Add CO2 or remove CO2? And then for this part, hydrogen carbonate absorb CO2 is not correct reagent because it should be the potassium hydroxide or the sodium hydroxide. And then for this part, I think that you would like to write down lime water because you say that it turns milky if respiration proceed. So in this setup, you choose the correct reagent. However, for the whole design, whole setup, it's not a valid method to show the change in the carbon dioxide concentration or the production of the carbon dioxide. So for the critical skills, it's necessary to keep the sample in the dark. It could carry out photosynthesis in the light, which would interfere the net change in the gas exchange. And it's also necessary to place the connect tubes correctly in the closed system. For example, for the tube, it should be immersed in the solution. And it's a closed system, the airflow will not be blocked if the connect tube are wrongly placed, and the gas will leak in or out of the setup if the system is opened. So beware of that. So I would like to recall one idea in gas exchange in human chapter. So do you still remember this setup? When we breathe in or out from the mouthpiece, so you can see that for the tube, immersed in the solution, it means that the air will go in. And for the green one, is not immersed in the solution, the air will go out. When you breathe out, the air should go to the right hand side test tube. And when you breathe in, the air should come from the left hand side test tube.